Okay, we were talking about fats and this concept of saturated and unsaturated fats. Um, um, the unsaturated fats are going to be liquid at room temperature, so you can think of something like oils, olive oil. And saturated fats will be solid at room temperatures, thinking of something like butter or um, I guess we'll stick with butter. Most people have never seen lard or nor know what it is anymore. Maybe you have. I don't know. Um, the thing about fat, it will separate from water because of the hydrogen bonds within each other. And so they generally a fat molecule is going to be hydrophobic, so it's not going to disassociate in water. We will talk a little bit about a phospholipid in a minute. It's a special consideration, but we'll get there. So the three fatty acids together, we did this on the last one, but by an ester linkage, creates a triglyceride. That's why I keep saying triglyceride, because the triacyclic glyceride is not a term that's often used. Triglyceride is the term that's used. The length of a number, they vary in length, the number of carbons, and they also vary in where those double carbon bonds are. The term hydrogenation is the process of turning unsaturated fats to saturated fats by adding a hydrogen. And this hydrogenation often happens in processed foods. And all of this sets up a scenario where it turns a fat that might have been healthy because of its unsaturated bonds into something that has a tendency to put something called plaque down in your particularly your coronary arteries. We call it atherosclerosis. So that plaquing in your coronary arteries that then can lead to cardiovascular disorders. You've probably heard about that. The other thing about just when I think I have a slide on something so I don't say it, is you also have this concept of trans fatty acids. So remember our structures, our geometric structure, you can have a cis and a trans depending on where the active group is. If the cis is on the same side or the trans on the opposite side, with a trans fatty acid, what they've done is they've made a double bond, so it's in theory an unsaturated acid, but they haven't made it they made it on the trans side. So the trans fatty acids are particularly unhealthy. Here we have our phospholipid, and even have an image of your bilayer sheet. This is the sheet or the arrangement of all the cells and cell membranes. We're going to talk about cells in the next chapter, so we'll talk about this even more so. So what happens is you have that same fatty acid, only now you only have two of them, and they're attached to a glycerol. That's the same thing. So you have two of them attached to a glycerol, and those fatty acids can be as long or short as is needed. But a phosphate group is also attached, and that phosphate group is going to carry a charge. So now with the phosphate group carrying a charge, that portion of the molecule becomes polar. And we know that water is attracted to polarity due to the oxygen and also the hydrogens. So what you have is this natural arrangement where the fatty acids will repel water or be repelled from water, and the phosphate will attract water. And you have two of them together where the phosphates will be on the outside. What you have is water outside the cell and water inside the cell, and this makes the perfect arrangement for the cell membrane. And this is, as I said, all of your cells have a membrane, and all of the cells that have membranes need lipids. Lipids are very important for you. Lipids also make up steroids. The one at the top are actually the testosterone and estrogen, probably looks familiar. The carbon skeleton of the 4 fused rings with the different active groups off of it are the difference between testosterone and estrogen. Also, your fat-soluble vitamins, vitamin D, is a steroidal ring, a series of steroidal rings. Cholesterol is a component, is the component of vitamin D in your what do I say? I just misspoke, didn't I? So according to your information here, steroids are the 4 fused rings, but you do have a steroid component to your testosterone and estrogen, as well as cholesterol is a component with cell membrane, as well as a precursor of sex hormones and vitamin D. And then you have the list of the sex hormones, the sex hormones written out there. Then that's it for lipids. Then we'll move on to protein. Protein is extremely important for the body. Many different functions protein gives us. As a matter of fact, according to your book, more than 50% of your dry mass, this is if they take the water out, more than 50% of your dry mass is proteins. And proteins, remember the monomer of proteins is an amino acid. Amino acid is going to be linked by a polypeptide bond. Proteins tend to be linear. They don't tend to branch. So they tend to be linear. And proteins are structural reports. So your muscles are protein based. You have the storage. Protein is functions in storage. Protein has the ability to store glucose, glycogen. Proteins are used for transport. If you want to attach something, actually, heme, the protein that's used to make blood. You can think of that as transport. Communication. These would be hormones. Are made of protein. Your antibodies are made of protein in terms of defense. And so the monomer is so designed where you have an amino group and a carboxyl group, and what you have is a carbon in the middle. You can see this on the left where you have this carbon in the middle. They call this the alpha carbon. Remember, the carbon has 4 bonding sites, so that carbon is bonded to the amino, the carboxyl, and a hydrogen. That is the same in every amino acid, and there are 20 different amino acids that are important for human life. So that amino, the hydrogen, and the carboxyl group are the same. It's the R group that's the variant. So the R group and what's bonded to that particular bond of the carbon is what makes the that particular amino acid. And what happens in terms of creating a polypeptide bond or a peptide bond, we can do it multiple times and then be poly, is you're going to do the same thing. You're going to take water out, so dehydration synthesis, and create a bond, create the peptide bond that way. And you have your backbone, so the backbone is going to be the amino, alpha carbon, carboxyl group, and then the side chains would be the active, the R group, the portion that actually is going to define that particular amino acid. Out of those 20, all of the proteins that are in your body are made. It's a sequence. It's a matter of how you are sequencing those 20, or how your body sequences those 20. The thing that actually isn't listed up here is your enzymes are protein based too, in terms of function. Here's a nice image in the overview of protein functions. You have enzymes. Enzymes are catalysts that will facilitate the reactions in the body without markedly changing themselves. Your antibodies are proteins. You have storage proteins, so milk protein, actually storage in egg white is a protein, and then transport proteins. So you have hemoglobin, as I talked about, and many other proteins that transport different things in the blood. Proteins are embedded in cell membranes to create channels. Here's your 20 polypeptides, the 20 amino acids. And you will not have to memorize all of them, so don't really worry about it. So understand that there are 20 amino acids. Some are nonpolar, some have a charge, and again I apologize for cutting that off. Some are polar, and some have a positive and a negative charge. So some have a positive and negative charge. That means they actually act like an acid or a base, so positive or negative charge. And then some are polar and nonpolar. So they're one or more polypeptides. They differ based on their R group, and the length can be a few amino acids to thousands. Generally what they are is they're linear, but you do have some hydrogen bonding, so that makes proteins three-dimensional. So the polypeptide is linear, and but those linear structures get together. Think of it like linear being the length of a piece of yarn is linear, but that yarn can be tied in knots, so it can actually make it a three-dimensional molecule, which most proteins that function in the body are three-dimensional. The making of the chain, we've kind of looked at this already. It's a dehydration synthesis. The carboxyl group attaches to the amino group, so we don't mess with the R portion, and we don't mess with the alpha carbon. So the alpha carbon and the R side chain are not affected. It's the carboxyl and the amino end that's that the bond is broken off and water is taken out. And then a little bit better picture of the protein structure. On the left you have the top, which is the stringing together the amino acids. That's considered the primary structure. And then what happens is those amino acids are pleated. This is because those hydrogens, if you remember correctly, have an affinity for one another. So the hydrogen bonding causes that string to kind of turn into a bit of a one of those the kind of a fan repeating pattern. And then the tertiary is it becomes the folding because even more interaction, and then you have the quaternary, that's a mouthful, is more than one acid chain getting together. So you have several, you can have at least more than one. So we start out with a line, we fold the line, then we actually create the three-dimensional, and then that three-dimensional can be among two of those polypeptide chains. The image on the right is basically showing you the different ways that proteins can be represented. You can represent it by the backbone, the wire, ribbon model, or a three-dimensional computer simulated model. They just different ways that the design of a protein can be shown, and we'll pick this up on the next one.